So this is Advanced Higher Chemistry, and this video is on Pharmaceutical Chemistry, which is a very small unit within Unit 2, but one that is always asked about in some form or another. Now, in Pharmaceutical Chemistry, what we're talking about are chemicals that are considered drugs and or medicines. So we need to know the distinguishing features between what is a drug and what is a medicine. Now, drug is the bigger category of chemicals. There are more drugs than there are medicines, because drugs are any substance that can alter or affect biochemical processes. So biochemical processes are anything to do with controlling your blood pressure, your pH of your blood, the chemical reactions that go on inside of cells, making proteins, breaking down sugars, and then also all of the neurological reactions that control mood, behavior, and your conscious thoughts, and either slowing that response or speeding it up. Now, drugs that have a beneficial effect on human functioning are what we call medicines. So all medicines are drugs, but not all drugs are medicines. To be a medicine, you must have a beneficial effect on the body. Now, when you buy a medicine or when you take a medicine, what you are consuming is not just the active ingredient, the thing that has the beneficial effect. There are other things that go into that pill, tablet, powder, capsule, etc. The biggest one is filler. Filler adds bulk. Now, you need to add bulk because most medicine doses are in milligrams. So you might be having something as small as 10 to 5 milligrams of active component, and a pill that size would be absolutely tiny. It would easily get lost and wouldn't really be useful. So it gets bulked out. The bulking agents can also help when you have consumed the medicine to allow it to be released more slowly into your body so that you can get a longer lasting response, or so that it's what we would call more bioavailable. Some medicines are in equilibrium in your bloodstream, and if it all gets released at once, only some of it will actually make it into the bloodstream and the rest would just pass through you. Whereas if you have slow release, you can allow that equilibrium to work in your favor. Another thing that can be added is sweetener. That's because the majority of medicines are made from chemicals that would really not taste nice. They would be exceptionally bitter. It can be very alkaline or very acidic. So sweeteners help cover the taste. And capsules, anything with like a shiny coating, that is sugar. Now, when it comes to medicines, how they behave, there's really only two ways in which they typically work. They are either going to act as an agonist or an antagonist, and you need to be able to give definitions for both of these words. So when medicines interact, what they are doing is they are binding to particular proteins in the body. The two most common types of protein they would be binding to are receptors. The receptors coat every single cell, and they're responsible for receiving information. Or they could also be interacting with enzymes, and as we know, enzymes are biological catalysts, which allow your biochemical reactions to happen at a rate that actually allows life to exist. Now, medicines will either interact in a way with the receptor or enzyme to turn on the natural response, or to inhibit the natural response. So if your drug mimics molecules that would normally bind to your enzyme or receptors, and therefore stimulate or mimic the natural biological response, that is what we call an agonist. So they promote the natural biological response. The opposite is preventing the natural response, blocking the natural response. That is an antagonist. Many drugs are enzyme inhibitors, and the part of the enzyme they would be interacting with is the active site, and not necessarily, for those of you who do biology, before anyone talks about allosteric sites. But if you block the active site, you will prevent the natural molecule from being able to enter the enzyme and do the reaction. So how do drugs function? Well, drugs interact with protein targets through intermolecular forces. So all the intermolecular forces from higher come into play within medicines. The active site or the receptor must be complementary to the molecule it's interacting with, the molecule it's binding with. So there may be regions of the active site or receptor that are ionic, that can form hydrogen bonds, or might be nonpolar and only form London dispersion forces. When a molecule binds to a receptor or to an enzyme, the ionic part must match up with the ionic part, etc. But not just that. If the receptor has a negative charge on it, the molecule that's binding to it must have a positive charge to create an attraction. So they must be complementary. If we know the structure of the active site, or we know the molecule that would normally bind to our receptor or our enzyme, we can design drugs that can have similar structural features and make similar types of bonds, which would therefore make them work more effectively. So how do we identify molecules that bind to targets? So the first thing we would do is you would go into the laboratory, or you would go through your library of compounds that your drug company has already made, and you would test them on the natural substrate, on the natural protein, the receptor or the enzyme, and you would look for which ones gave you a response and which ones didn't. Now, once you have found these molecules all give a response, you would look for what are the similar structural features within all these molecules that have been successful, and therefore that allows you to piece together what sort of intermolecular forces you need to be able to generate, and what maybe particular bonds you need in what sequence in order to make this molecule. So here's an example that you could get in an advanced higher test. They usually give you between 4 and 8 molecules and tell you the ones that are active, the ones that are inactive. In this example, what we have are the ones on the left were all active, and the ones on the right were all inactive. Now, what you need to do in a question like this is look at all the ones that are active and look at absolutely every atom that they have in common with one another. Those atoms that are identical in all the active drugs are the region that is binding. So if we look at the molecules on the left, we can see they all have an NH2 group at the left-hand side. They then have that benzene ring, they have an SO2, and they have an NH bond, an NH, and then they have something else bonded on the right-hand side. Now, all 3 of those molecules on the left, the right-hand part of them is all different, but we know that they're all still active. So what that means is the thing on the right end of the molecule isn't important. It's not involved in binding. Now, when we look at the ones on the left, we can see that of this structural feature that we had on all the molecules on the left, something is different. So that top molecule, we see that instead of an NH2, we've got a CH3 group. That CH3 group has interrupted this part that is necessary. In the second molecule down on the right-hand side, the second nitrogen, the one on the right-hand side, well, it has a CH3 group where all the ones on the left have a hydrogen. So that is interfering with the way that it can interact. And the bottom molecule doesn't have that second nitrogen, it has an oxygen instead, which is telling you that it's not capable of forming the exact same type of bonds and it's getting in the way. What this leads us to is our binding unit. So that's the bit I shaded in on purple. So this is our binding unit. Now, once we've identified this, we can look at what sort of intermolecular forces are probably involved in the receptor or active site and build even more molecules. So the NH groups are capable of hydrogen bonds. The hydrogens themselves will be able to interact with delta negatively charged things on the active site or receptor. The nitrogen is delta negative, so it will be able to interact with delta positive things. The SO group is polar, and where you've got an oxygen on its own, it can accept hydrogen bonds. So if something on the active site has an NH or an OH group, that hydrogen can interact with those oxygens. The benzene ring, well, benzene only contains carbon and hydrogen, so it is very non-polar. So we've got a whole middle region where we need to be able to make London dispersion forces. And as I said, that bit on the right didn't seem to be important when it came to binding to the molecule. And so that gives you scope to actually introduce variation and see if by changing this part, you can maybe increase efficacy, so how effective it is, or maybe find an even better set of groups by extending it that could bind more effectively. So it's by investigating molecules that do work, you can then learn more about the target and about how to make a more effective medicine.